what will happen is all these protons are going to come back through the ATP synthase. So this is the proton kind of a channel for the excess protons that are there. You can imagine this as an H plus turbine, not like a water turbine, but like an H plus turbine. So turbine, what does that do? It has like a rotating shaft kind of a thing, in which when you put water on that, it starts to rotate. This thing will rotate because of the H plus ions. While it does that, this converts, so this will rotate and this will convert the ADPs into ATPs because of this rotation. This is the process where most of that energy is produced. So electron dumping, protons getting accumulated on one side. That proton, through this ATP synthase enzyme, is rotating this enzyme. Because of that rotations, the ADPs and the phosphate groups align themselves in a particular way, and this alignment and this rotation starts producing a lot of the ATPs. About 32 to 34 ATPs can be produced in this step. Now, just to recap the different processes and the energies that are being produced. So we can write the different stages of aerobic respiration and the number of ATPs produced, the net number of ATPs, right? The net ATP. So glycolysis produces 2. The pyruvate to the acetyl CoA produces 0. The Krebs or the TCA cycle produces 2, and the final electron transport chain and the chemiosmosis, so this process that we just discussed, produces 32 to 34 ATPs. So this is aerobic respiration. Now, why is aerobic respiration important? So any complicated life form will need a lot of energy. We need this kind of like aerobic respiration process for a complicated life form because of the amount of energy that is required. Okay? This is much more efficient in producing the ATPs, because if the idea is to produce ATPs, this is a much better, much more efficient way of producing the ATPs. Okay? Now, moving to anaerobic respiration. What would be common in aerobic and anaerobic respiration? The first step is the common. Now, there could be 2 types of anaerobic respiration. One we'll call it fermentation. The other I'll call it true anaerobic respiration. The second part is easier. The true anaerobic respiration. But let's look into the fermentation part. So we already saw that one thing was common. The first step is the same. So in fermentation, when you have the glycolysis, so you start with the sugar, same, you start with glucose, you produce the pyruvate, and you produce 2 ATPs, correct? This is the same. And what other type of battery did we produce? NADH. How did we produce this? So we recharged this NAD plus to NADH. When do you think, so you can still produce 2 ATPs, right? So if you have a lot of this glucose, even when you don't have oxygen, so oxygen is not required in this step. So even without the oxygen, you are able to produce ATPs, right? So why can't you just do this always and just keep on producing these 2 ATPs? Is it possible? Yes, this is net 2 production. Yeah. But if you don't have anything else, can't you just keep on producing this?
Okay, those batteries, charged batteries are produced, so electron has to be goes up.
Yeah, so electron are producing. So this is the charged battery.
Yes, so electron.
Can't you just do this forever? Glycolysis. Can you keep on doing glycolysis until this sugar is gone? Can you do this? Just convert this glucose into pyruvate forever until that glucose is over. Because at least you are producing 2 ATPs. But this does not happen. Why? Because this is in a limited supply. For this process to happen, you definitely need someone to take up your electrons. If you don't have anyone to take up your electrons, your reaction will stop. It will not. The reaction will say, hey, I need more people to dump my electrons to. Give me more of them. But if all of your batteries are now charged, well, where will you get those batteries? This is the holdup here in glycolysis. The limited supply of NADH is the holdup. Now, you need this NADH to be reduced back to NAD plus to keep this glycolysis going. So someone needs to take those NADH and then again convert it into NAD plus. Now, this is the second part of the fermentation process. Okay. Now, the glycolysis will be same for both of them. You can see that glycolysis is same for aerobic and anaerobic, all these kinds. Now, the second process in fermentation will actually take that pyruvate and do something with the pyruvate. So the second step will take that pyruvate and do a few things. Pyruvate or pyruvic acid. What is the difference between pyruvate and pyruvic acid?
Pyruvate is salt.
Right, so you just remove the H from an acid, it gets the name 8. For example, sulfuric acid, sulfate. right? So this is the lactate, lactic acid. Just remove the H plus. So pyruvate, when you remove the H plus, you get the so you get this pyruvic acid or pyruvate. Then this is going to do a few things. So most importantly, this can be converted into alcohol, or this can also be converted into lactic acid. In this process, what happens? You end up using the pyruvic acid or the pyruvate that is being produced, but this thing also produces a little bit energy. It also produces 2 ATPs. This process also creates NAD+. Now, because you have discharged the NADH again, now this NADH can again go and keep the glycolysis running. So between this and this, you can still produce 2 ATPs and 2 ATPs. That is what is happening if you don't have any oxygen. So if you have oxygen, you end up producing a lot, 32, 34, whatever. But when you don't have, you're stuck with the glycolysis and this stage of anaerobic respiration. This is the fermentation stage. So this is the fermentation stage, when pyruvic acid can either go into alcohol or can go into lactic acid. So anytime you produce alcohol, this is the process that you are actually forcing things to do. Any kind of alcohol, beer, wine, the whole alcohol industry, this is the process that they do. Okay? There has to be no oxygen. You take a sugar or any starch or any sugar source. Now, depending upon different sugar sources and the different complexities, you will have different flavors or different types of the alcohols, right? So this is essentially the alcohol production business. This is what happens to your body, right? Also happens to curd, yogurt, right? So like this is the lactic acid that is being produced. Cheese, this is what is happening in cheese. That is what is happening in alcohol. This also happens in your body. So when you run for a long period of time or you're exhausted, you end up, if you're not breathing well, okay, so you're short breaths and doing this, then you end up producing a lot of lactic acid in your body, and this is the thing that hurts. So any of the cramps and any of like the pain that you have after a intense session, in which you're not breathing well, this is the pain, the lactic acid. Okay. Then when you again dip your feet in warm water, why does the pain go away? This is breaking down the lactic acid. That's the pain going away, the breakdown of these things. Okay. Now, the end product of the alcohol is the alcohol, which is like typically is an ethanol, C2H5OH, and in here is the lactic acid, of course, right? Coming to the true anaerobic respiration. In this, we are only changing the electron acceptors. So in this case, we will just have different electron acceptors and not oxygen. So anything that can accept those electrons can actually behave as, can actually induce the true anaerobic respiration. The energy produced in this will be most likely be higher than the fermentation process. Because you have to understand, in anaerobic respiration, The only thing that is changing is the electron dump. So entire process remains the same. So all that protein complexes that we saw here, everything remains the same in the true anaerobic respiration, except the fact that oxygen is the terminal electron acceptor and in the aerobic respiration, in here you will have something else. That's the difference. So this is the true anaerobic respiration. This is fermentation. Really less energy production in here, higher energy production in here. The highest energy production is typically observed in aerobic, but we'll just compare that in a bit. So there will be organisms that can actually use nitrate as the terminal. So there is this Pseudomonas bacteria will actually use nitrate as the electron acceptor. What will happen to this nitrate? It will get reduced. It might produce what? It can produce nitrite, NO2 minus. What else? I don't know exactly what this organism will do, but yes, nitrite or NO, N2O, ammonia, nitrogen gas, all these reduced nitrogen species can be produced. So this is the most oxidized species. You can end up producing a lot of these reduced nitrogen species. For the sulfur, there is a class of bacteria called D-sulfovibrio. I don't know these names or anything, but just for your sake, you take the sulfate, this is the kind of bacteria that can do. What will they produce?
Sulfite.
You can produce sulfite, yes, but as we discussed in the last class, that because the sulfur is in what oxidation state here? +6, right? So there is a long way to go from that. So you can have a +4, which is the sulfite, right? There could be some other bacteria that are present there who can take those intermediates and then reduce that, maybe produce something else, take that, produce something else. What would be the terminal thing that is being produced? What is the last thing that you think this is going to get reduced to? H2S. Right? So H2S will be produced in these. And H2S has a peculiar smell. What is that smell? Rotten eggs. So it actually smells like rotten eggs. So that rotten egg smell is because of the hydrogen sulfide gas. Okay. Now, it could be abiotic or biotic hydrogen sulfide, but in this case, you typically get hydrogen sulfide gas. Okay. Now, if you compare the three different pathways, right? So you have aerobic respiration, anaerobic respiration, or the fermentation or the true anaerobic respiration. Where do you need the oxygen? Only aerobic. You do not need the oxygen in fermentation. You do not need the oxygen in the true anaerobic, right? Does it include glycolysis? So let's write this. So aerobic, in here let's write fermentation, true anaerobic. Is the oxygen needed? Yes. No. No. Do you have glycolysis? Yes. 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 Do you have the Krebs cycle? Yes. No. No. Electron transport chain? Does this have an electron transport chain? Aerobic? Does this one have electron transport chain? No. Does this one have? Yes. Final acceptor is what? O2? What is the final acceptor? The pyruvate, because this is where it stops. This is the last one that can actually take the electrons. That's why you don't have a lot of electron acceptors. In here, any number of things, right? NO3, sulfate, perchlorate. Net ATP yield, (36 to 38), but there will be some problems with it. Won't be as efficient. It also depends on human being or the organism that is doing it, right? What is the net ATP yield of the fermentation? In the glycolysis 2, and then in the fermentation process 2. So I'll just write 2 in the fermentation process. I mean 2 plus 2, let's write it this. And true aerobic, it is less than the aerobic, but more than the fermentation. But anywhere in the range of 5 to 30 ATP. It depends on how good these electron acceptors are. If you have a very good electron acceptor, then you get more ATPs. What is the final product of aerobic? Here? Alcohol or lactic acid? Here? Reduced form of these. So it could be variable, right? So you can get NO2 minus NO. You can have so 3,2 minus. I also saw somewhere these. And finally H2S. Perchlorate can actually give you chlorate, probably, I don't know, maybe hypochlorite, chlorite, even maybe chloride. Okay, so all these things. So does this give you a decent idea about this whole energy business? Now we'll have to also understand that these may be better electron acceptors or maybe less efficient electron acceptors. It will depend on the oxygen, right? Some of these are, why should we care about these kinds of things? I get why should we care about the aerobic, because we after all are doing aerobic respiration. We are complex organisms, we need a lot of energy. So this is what we will do. I get the fermentation process because when you don't have a lot of that, at least it gives you something to work on. And there is a big food industry that benefits from that. Lactic acid, cheese, bakery, alcohol, all that. What is the benefit of this thing? Why are we even concerned about this? This was happening before we had oxygen in our atmosphere. So when Earth did not have an oxygenated atmosphere, there were still organisms living, right? This is what they were using as the electron acceptors. In absence of oxygen, what can you do? You don't have the oxygen. So that is another reason that we did not have a lot of complicated life forms before the advent of oxygen. The advent, when you have a lot of oxygen in the atmosphere, you start having aerobic respiration. That is a reason that we can afford complicated lifestyles. Otherwise, you cannot have a complicated lifestyle. If you have a huge energy need and you don't have that energy, how will you? A primary difference between, first world countries and the developing countries is this: the energy requirement. First world countries the developed countries will have a lot of energy requirement, and they need to support that a lot of energy requirements with a lot of things. On the other hand, the developing countries, they do not have a lot of energy requirement. They need it, but they don't have it. So the lack of that energy is actually keeping them behind. So they don't have the electricity. They want to sit quietly in a room and study, but it's sweltering hot and there is no electricity. So they end up not studying. It's a very naive example. But that's what, the energy requirement. So in here, when you have a lot of energy requirement for this, and you don't have O2 or something like a good electron acceptor, you end up just sustaining. There is less development, there is less complication. That's how you stay there for a long period of time. Only when you have a lot of oxygen, then you can become, okay, hey, we have this electron acceptor that we can tune our bodies to, and then we can have higher and higher ATP production. Now that we have a great amount of ATP production, now we can afford a lot of these complicated lifestyles. Okay? All right. Now, this was the reason to understand these. So this was all about the aerobic versus anaerobic. So this is all about the electron acceptors. Coming back to the electron donors. In here, in all these three, you must have seen the electron donors were the organic carbon. So you always started with the glucose in here. We were always starting with glucose. So this is a reduced form of carbon. Now, there will be organisms that can extract the energy from inorganic electron donors as well. For example, carbon dioxide. So these are the phototrophs, right? So these are the autotrophs, right? In which you can take that CO2 and convert it into, and use that as the electron source. You could also have iron 2 as the energy. So in a place where you don't have organic carbon or you don't have CO2, if you have iron 2, some organisms can actually use that, and they will convert this into iron 3 plus the electron, and they will utilize this electron to do their business. Now you can imagine this could be many things. A lot of things can actually donate these electrons. However, what other things can you have as an electron donor? Anything that is reduced, think of that. Anything that is, yes. So if you have a sulfide, so HS minus, something like that. Or methane, this is also reduced. So these could start behaving as the electron donors. Now,